right, hi everyone. We're moving on to our next chapter, which is microscopy and staining. I thought I would just start by showing you this slide with the sizes of different organisms. Some are macroscopic, right, like the dog, and others are microscopic. There are two main types of microscopes, the light microscope and the electron microscope. We'll see that the resolution that we get with the microscope is based on the wavelength of the illuminating source. And with the scanning electron and transmission electron microscopes, their illuminating source is the electron, which has a much shorter wavelength, which means that you can see much smaller organisms and much more definition of detail, because you are resolving smaller structures than you can with the light microscope. So with the light microscope, light is the illuminating source, and it has a much bigger wavelength than the electron. So the things that we can see with our compound light microscope, basically we can see human cells like red blood cells, we can see bacteria, we can see some of the larger organelles like the chloroplast and the mitochondria, but we can't see viruses. Okay, so I think I mentioned in the last lecture that some viruses can be like 20 nanometers in diameter. That would be much smaller than something that we could see with our compound light microscopes, and I'll talk about resolution in just a minute. What's interesting with the electron microscope, specifically the transmission electron microscope, is that we can see almost everything with no size limitation. So if you look, we can see the diameter of DNA, we can see amino acids, almost down to the level of the atom. So the transmission electron microscope almost has no size limitation. So I just wanted to speak about resolution for a minute, and I'm sorry that this equation for resolution is written that way. I should really probably put parentheses around 2 times the numerical aperture, because it's the wavelength lambda divided by 2 times the numerical aperture. Like that whole 2 times the numerical aperture is in the denominator. So resolution, or the R value, or the resolving power of a microscope, is in general improved when the wavelength of the illuminating source is smaller. Okay, so the smaller the wavelength, or the shorter the wavelength of the illuminating source, the greater the resolution. So you want the resolution basically to be a small number. And what that means is that if something, like if the resolution of a microscope is 0.1 micrometers, that means that if 2 points that you are trying to resolve, okay, or separate, are 0.1 micrometers apart, that microscope will be able to resolve them, distinguish between them. But if they're closer together than that, okay, then they are not going to be resolved or separated, and they will look like what you see in part B of this figure. Okay, so that would be something that would fall below the resolution of the microscope. Don't get hung up too much on the numerical aperture. This is actually a value that is engraved on each of the objective lenses of your microscope. And so you can actually calculate the resolution for each of the objective lenses on your microscope by finding that value and then plugging it into this equation and taking an average of the wavelength of visible light, and you can calculate an R value. I'll talk about the R values for the oil immersion objective and when we're talking about the features of the different microscopes. So in this figure, we're looking at wavelength, okay, and it's showing the spectrum of colors of different wavelength of visible light. And like I said, you could take the average of that. Okay, so they have the average wavelength written right there. It's 0.55 micrometers or 550 nanometers. And that would be what you would plug into that equation. R equals lambda over 2 times the numerical aperture. So what you can see is that visible light is this spectrum. So it starts with, on the kind of the blue end of the spectrum, you have very short wavelengths, and then going to the red end of the spectrum, the wavelengths get longer. Okay, but we usually average the wavelength of visible light, and that would be what we would plug into our equation. I just want to mention that there is one light microscope that uses UV light, and you can see on this figure that the wavelength of UV light is a little bit shorter. And so that would make the R value a little bit smaller, right? So the resolution would be a little bit better with a microscope that is using UV light instead of visible light. I like this figure because it shows the relationship between wavelength and resolution. So remember, resolution equals the wavelength over 2 times the numerical aperture. So the R is equal to the wavelength, and when the wavelength gets smaller, the R value gets smaller. Okay, and when the R value is smaller, the resolution is better, right? Okay, so if the balls and the beans and the beads are representing the wavelength, you can see on the other side of the figure that you have the resolution, and as those balls and beans and beads are getting smaller, okay, as wavelength is getting smaller, your resolution is increased, and you're getting this nice figure with increased definition and clarity of detail. Okay, just to mention some light properties. So you can see in the figure reflection. So light could be reflected off the surface of a specimen, or light could be transmitted through a specimen. All right, we'll see with the compound light microscopes that light is actually transmitted through our specimen and collected by the objective lens. Light can be absorbed at one wavelength and transmitted at a different wavelength with fluorescence, all right? And that's what's happening with the UV light microscope. And then light can be refracted, okay, or bent, all right? So you can see that if, say, for instance, the specimen has a different refractive index than the air, okay, then the light would be bent, okay, or refracted. This is refraction, where water and air actually have different indices of refraction, and so it seems as though the pencil is broken. But if you were to pull the pencil out of the water, you would find that the pencil is intact. Okay, so this is an optical illusion, but it has to do with the index of refraction and how those two indices between water and air are different, okay? And the index of refraction is defined as the speed at which light passes through a material. I just want to mention, and we won't necessarily be in the lab, but when you're doing microscopy and you use the oil immersion objective, the reason why you add oil to your specimen is because you're trying to decrease refraction of light. So you want all of the light that is coming from the illuminator up through your specimen to be collected by the objective lens. But if your light comes up through your specimen, okay, and the light is coming from a glass lens, which is the condenser, up through your specimen, which is on a glass microscope slide, and then it hits the air before it's collected by the glass objective lens. The air and the glass would have different indices of refraction, and the light would actually be bent and could be lost and not collected by the objective lens because it's bent and moving in a different direction. And that would actually decrease the resolution of your image. But if we add oil to the top of your specimen that is on the microscope slide on the stage, and the objective lens actually goes in and touches the oil, okay, and the oil has the same refractive index as the glass that your specimen is on the microscope slide, and also the glass of the objective lens, then all of that light would move straight into, without refraction, straight into the objective lens, and none of it would be lost due to refraction. So this is an image of a compound light microscope. Light is the illuminating source. It's a compound microscope because it has a system of magnifying lenses. It has the ocular lens, which magnifies things tenfold. And then it also has the objective lens, which is on a revolving turret. Okay, and basically depending on which objective lens you're using, okay, if you're using the 4x lens or the 10x lens or the 40x lens or the 100x lens, you will magnify the image however many fold. Okay, when we talk about the features of a microscope, okay, the two features of a useful microscope are magnification and resolution. The magnification, the highest magnification you could achieve with the compound light microscope would be 1,000-fold magnification. And with the oil immersion objective, the resolution that you would get would be about 0.2 micrometers. Okay, so basically, if we were in the lab, we could look at the objective lens for oil, okay, the 100x objective, and on the side of that objective lens, we would see engraved the number for the numerical aperture, which would be 1.25. So if we plugged that into our equation, R equals wavelength over 2 times the numerical aperture, it would be R equals 550 nanometers or 0.55 micrometers divided by 2 times 1.25. Your answer is about 0.2 micrometers. Okay, and so that is the R value. Okay, so basically what that means is that if 2 points are 0.2 micrometers apart, they can be distinguished, okay, or resolved. If they are closer together than that, then they would not be resolved, okay? So they would be like, you know, part B of that figure where you're just seeing the giant blob. Okay, these are some images from the bright field microscope. Light is transmitted through the specimen. The specimen appears on a bright field. And so usually what you need to do is stain your microbe. Okay, so these microbes are all stained. If they weren't stained, okay, you would have a difficult time seeing them. Okay, it's not impossible to see them, but it is a lot more difficult to see them. So to provide contrast, the specimen is stained, okay? And to do the staining, where you're adding stain and you're, rinsing your slide and etc., you need to fix the microbes to the slide, okay? And usually we do a heat fix or a methanol fix, and that adheres the microbes to the slide, but it also kills them. And then we add the dye, okay, and we rinse it off, and we can look at the microbes under the microscope, because now there's this contrast. And this is actually the main use of your bright field microscope is looking at classically fixed and stained microbes. You can look at viable or living microbes with the compound light microscope, but since they're living, okay, and they're not stained, you're going to have trouble seeing them. And so I wrote up there that the limitation would be poor image production. This is the dark field microscope, which is, actually another type of light microscopy. Okay, so the features, all right, would be exactly the same. Okay, the magnification and the resolution. With the dark field microscope, light is not transmitted through the specimen. It's reflected off the specimen, and the specimen appears on a dark field. Okay, so you can see in this figure, some yeast, and they kind of almost look like they're glowing, right? So the condenser contains an opaque disc, and actually, if we had fancy microscopes, we could have different condensers. We could have like a disc below the stage where we could just turn that disc and go from bright field to dark field by switching out the condenser, and we could do dark field microscopy like this. Dark field microscopy is usually used when you have microbes, and maybe they're even, microbes taken from a patient that cannot easily be cultivated in vitro, in the test tube. So you want to maybe take that sample from the patient and just look at it under the dark field microscope. It used to be that every clinic had a dark field microscope because it was used in the diagnosis of syphilis. Syphilis is caused by a spirochete called Treponema pallidum, and Treponema pallidum is not easily cultivated in vitro. But if you took a sample and you looked at it under the dark field microscope, spirochetes are mottled by endoflagella, and so they have kind of an erratic, jerky movement. So if you saw a spirochete and it was kind of moving in an erratic, jerky way, that would be at least a preliminary diagnosis of syphilis. Okay, so this microscope is not really used for fixed and stained microbes. Okay. All right. Another type of light microscopy, which would have the same features, the same magnification and resolution as the bright field and dark field microscopes, would be the phase contrast microscope. The phase contrast microscope has a special condenser, kind of like the dark field microscope. And in fact, if we had a fancy microscope, we could, have a disc that we could turn and, put that special, that special phase contrast condenser in place, and we could do phase contrast on our light microscopes. Basically, light is coming from two sources. One source would be directly from the illuminating source, and then the other source would be from light that is reflected or diffracted off a structure of the specimen. So when those two sets of rays are brought back together, if the wave, think of like a bell-shaped curve, if the light from those two different sources is in phase, so they're both are overlaying each other perfectly, that would lead to reinforcement or increased brightness. But if they are out of phase, so like when one wave is peaking, the other one is in the deepest part of its valley, that is referred to as interference, and that is going to lead to decreased brightness or relative darkness. And what happens when you have these really bright parts of the image and really dark parts of the image is that you get a very highly contrasted image of internal structures. And so with the amoeba right here, we're seeing, using phase contrast, we're seeing a very, very highly contrasted image, and we're seeing a lot of definition of its internal structures. Okay, so this would be something that would be used on viable microbes, which are not stained. Okay, so they're living, all right, they're not stained. Staining would you would kill them in the staining process, and it's to see internal structures. Okay, so if you think about bacteria and prokaryotes not really having any membrane-bound organelles or a membrane-bound nucleus, this is not, a type of microscope that we would use a lot to look at bacteria. Okay, but it's really fun to look at the protists using the phase contrast microscope. The ultraviolet light microscope has slightly different features, okay, which we'll talk about in a minute. And that's because it's using UV light instead of visible light, so the resolution is a little bit better. This figure right here is showing some microbes that have been stained with a fluorescent dye called acridine orange. And acridine orange binds to nucleic acids, and depending on whether the cell is living or dead, it's going to fluoresce a different color. So live cells fluoresce this bright green or yellow color, and dead cells fluoresce a red color. Okay, so that is using a fluorescent dye to attach to the nucleic acids to determine whether cells are living or dead. So, okay, so this is considered light microscopy. It's using UV light as the illuminating source. The magnification would still be about 1,000-fold. But the resolution is 0.1 micrometers. Okay, that's a little bit better than the other 3 microscopes we saw. And I say there, why? Okay, but hopefully you have been following along and you realize that that's because we're using ultraviolet light, which has a shorter wavelength. Okay, and that's going to give you a better resolution. So the uses, okay, we saw where, microbes could bind selectively to certain dyes, like the acridine orange. There are some microbes that are naturally fluorescent, okay, like Pseudomonas aeruginosa is naturally fluorescent. Okay, we could just look at those under the UV light microscope. But the major use for this microscope is immunofluorescence. Okay, so immunofluorescence is where you have antibodies, okay, and oftentimes they're monoclonal antibodies that have been raised against, say for instance, syphilis, okay, and the organism that was infected with the syphilis wasn't necessarily even infected with virulent syphilis. Okay, it could be weakened, it could be acellular, but all right, they have raised an immune response, and they've raised antibodies against that, and then those antibodies can be harvested. Blood could be drawn from, say for instance, a rabbit, and antibodies could be isolated and attached to a fluorescent dye. Okay, and so now if somebody is suspected of having syphilis, that the sample from that patient could be put on a slide, and if the antibodies, which are highly specific, attach to that organism, and you look under the UV light microscope and you see a fluorescent spirochete, okay, then that would be a positive diagnosis of syphilis. Okay, rabies is also diagnosed in this way. All right, I'm gonna stop right there, and we will discuss electron microscopy in part B of this lecture.